Okay, hi everyone, and welcome to our HEAS talk in Ancient Genomics. And today I have the pleasure to introduce Professor Anna Szécsényi-Nagy from the director of the Institute of Archaeogenomics Hungarian Research Network, Research Center for the Humanities. And she's going to talk about genomic science of community organization.
Thank you very much for the introduction. So I would shortly introduce you my institute, which is within the Research Center of the Humanities. We are part of the Hungarian Research Network for 5 years now, and we are working on ancient human populations mostly, so ancient DNA analysis of these populations from the Carpathian Basin, but also from specific regions of Eurasia, from the prehistoric to the modern times. And our specific focus is the early history of the Hungarians from the Volga, Ural region settlement areas, and also from other areas along the way to the Carpathian Basin. But on the other side, we initiated recently archaeogenetic analysis of some domesticated animals, and now are developing new directions toward environmental and microbial genetics. Today's talk has the following outline. So I will first give you an intro about the background of this late Neolithic, early Copper Age period of the Carpathian Basin. And then I will present you some sites from our research study, present you some archaeological hypotheses that we are dealing with, and then we'll talk about actual and the new genetic results from this project and highlight our future aims. Just a few slides about my personal background. I did my PhD in Mainz, Germany, under the supervision of Kurt Alt and Esther Banfi. That was a joint, huge research project. German Research Foundation funded it. And we have sampled during this project around 70 sites from the Carpathian Basin. We did most uniparental analysis at that time, from the 6th to the 4th millennium BC period. And when I returned to Hungary from Mainz in 2013, and after some maternity leave from 15, I managed to collaborate with the team of David Reich from Boston, and together with his postdoc, Mark Lipson, we managed to reanalyze some parts of this sample set for full genomes. So 1240K capture was did at Boston, but the DNA libraries were prepared in Budapest. And we had this paper in Nature in 2017, where 3 model regions were analyzed from Europe, and from Hungary we had a larger sample set, 100 samples. And basically what we found is that there was an initial admixture between hunter-gatherers and the incoming farmers from Anatolia. And after this initial pulse event, we have a continuous admixture between the descendants of the foragers and the farmers. So a steady increase of this hunter-gatherer ancestry was observed through the studied period in the area. Another important reference data for the study that I'm presenting today was coming also from Vienna and the Harvard laboratory. So Pere Gelabert and this team published this paper last year. And what is important for us here now is this site Polgár Ferencihat from the Great Hungarian Plain here. Which is dated to the Middle Neolithic period of the area, to the ALPC, so Alföld-Linár pottery culture. This is a settlement site, and some individuals were buried between the houses here. And the team managed to analyze 45 individuals. Together, I put here some photos of colleagues from the ELTE University, so Hungarian colleagues who participated in this project. And a significant result from our aspect of this study was that Pere found a significant within community variation in the Western hunter-gatherer ancestry of these people. Within these genomes, and in general, he found patrilocality within the site, and also some families were described from here. Okay. Now I would like to shortly introduce you a huge Hungarian-based and Hungarian MTA is the Hungarian Academy. So Hungarian Academy founded project of Zsuzsanna Siklósi. You see her picture here, which she started 2.5 years ago. So basically, this project focuses on individuals and communities, social networks, and the spread of innovations in the cooperation of the Carpathian Basin. And her aim, or the aim of the team, is to reveal the various relationships between individuals, between larger social integration units and communities. So the team is using a multiscalar approach. And the methods within these projects are the analysis of material culture, anthropology, molecular bioarchaeology, and zooarchaeology. So I joined this team some years ago, and we decided with Zsuzsa to help this previously formulated project and collaboration to publish 125 individuals that were sampled previously from the Carpathian Basin by Tamás Hajdú and Ron Pinazi. So they initiated this project some years ago, and then we joined with Zsuzsa, who actually also gave some samples into this project. And that's what I'm going to present you today. So within this project, the laboratory work was mostly done in Vienna and Boston, and the analysis was performed in Budapest. This is now in a preprint form, what I'm presenting you. So basically, the background of this whole story is that we see that we experience, or the archaeologists hypothesize, a significant social change within a time span of 100 and 150 years in the Carpathian Basin. So it was around 4,500, 4,350 Cal BC. And what they detect is that many aspects of the life changed during that time. For example, new house types appeared, new settlement types appeared, formal cemeteries appeared. So previously we had burials within the cemeteries, and the dense network of small farmsteads were established or formulated. And the pottery style also became more homogeneous. And the archaeologists had several studies on the potential reasons behind these changes. And one idea is this scalar stress. So basically, the population increased so much that it culminated in the abandonment of these larger tell settlements and a shift to a smaller dispersed settlement structure. And it's important to emphasize that between this this time span of 4,450, 4,350, there was a kind of gap of radiocarbon data in the area. So it looked like as the population would somehow change or shifted or decreased and then increased again. In this study, we had several sites. Here they are marked with black dots, and used in the analysis some comparative data sets from the dots in gray. Our main focus area is the so-called Polgar microregion. So it is here on the map where we see the numbers from 8 to 11. This is basically a small area close to the town Polgar, and the ELTE University conducts here excavations and geological surveys and different kinds of work for several decades now. The other important site that I will present you is here, the late Neolithic site from Aszód, and one that I will mention several times is here from Szécsényi, close to the Hungarian border, but from Romania. They are all on the Great Hungarian Plain, so this green area here is the Great Hungarian Plain. Okay, so the first site that I would like to shortly introduce you is Aszód-Papi földek, so it is the number 2 on this small map. 224 graves were excavated here in the last century, and this belongs to the late Neolithic period of the area. The Lengyel complex, it was a huge archaeological culture spread over here from Transdanubia, from west of the Danube, and 22 samples were analyzed in the study. The preservation wasn't really good, so I won't really highlight this site anymore during my analysis, but you will see them on my plots. What we found here is just 2 small families, 2 pedigrees, closely related individuals were buried near each other. Unfortunately, the preservation wasn't really good, as I mentioned. For the deeper analysis, I will show you. A more important site from our aspect is from the Polgar microregion, late Neolithic period, Polgar Csőszhalom. It's a very famous site. You might have heard about it. There was a stele settlement here, so a multi-layer settlement with burnt houses and surrounded by circular ditch system. And there, due to motorway excavations, we had the chance to uncover a horizontal settlement next to the Tel settlement, which was even connected with the road. The two settlements were connected with the road. And due to several surveys, the archaeologists could define the total area of the site. It was almost or over 60 hectares. And the archaeologists presume that 400 houses could simultaneously exist in this area. So it was a huge settlement complex. And they presume that simultaneously almost 2,000 people could live here. And they mostly used, or probably used, this Tel settlement for ritual purposes, whereas they lived on the flat settlement area. And here on this surface, 80 houses were excavated and 123 graves. Out of them, 30 was part of this project. So it's from the late Neolithic of the period. And there is a time gap after that, as I told you before. So a shift to the early Copper Age, settlement structure changed. And in the early Copper Age, 2 cultures needs to be named. One is this Tisza-Polgár, the other Bodrog-Keresztúr. They were previously considered as subsequent periods, so strictly chronological periods and like monoliths of cultures. And nowadays we see it differently. We think that they were rather pottery styles and not cultures, and they were also overlapping. So due to the dense radiocarbon sampling conducted by the team of the ELTE University, now we do know that there was 130 to 130 years overlap between the 2 pottery styles. You see here the spread of the two styles in the Eastern Carpathian Basin. So going back to the Polgar area from this early Copper Age period, we have a very important and interesting site that we were lucky enough to get involved into the project. It's called Tisza-Polgár Basatanya, was excavated in the 1950s by Ida Kucian. And you see here different colors. These different colors are the different pottery styles I mentioned before. So the Tisza-polgár style and the Bodrog-keresztúr style. And there are some transitional graves. And previously it was considered as the subsequent subsequent burials of the cemetery. So chronologically different parts of the cemetery. And inherently, we have the question whether these were used by two different populations or whether they were interconnected. What was the chronology of this site from the genetic point of view, although we have radiocarbon dates, as you see here, and then we know now that the span of the usage of the cemetery was between 130, 300 years. Okay, I need to remember you that this site has a time gap between the other polgár area site, Csőszhalom, which I presented shortly before. And there is another small site here where we have a few graves also included in this project. I will come back to that later on from Polgár Nagykasziba, just a few graves around 10 kilometers away from Basatanya. Okay, the last site I would like to show you is from here, from the Romanian-Hungarian border with number 14. This was already analyzed for the Josif Lazaridis Southern Arc paper in 2022. But now we have new data from this site. So already 74 individuals are sequenced out of this 129 Copper Age graves. And it also has a time span similar to Basatanya, 140, 300 years, so almost overlapping, contemporaneous with this Basatanya site I showed you previously. Okay, and so one is here, Polgár area, and Uzsicsenyi here. There is around 100-kilometer distance between the two. Okay, so what were our goals? We wanted to understand a little bit better this late Neolithic early Copper Age transformation, so internal transformation within the Polgár microregion, but also in general, whether there was any kinds of mobility or what was the role of the cultural contracts in that period and era. We would like to get some genetic evidence about community organization and structure. And of course, we wanted to compare these two sites 100 kilometers apart, so Basatanya and Uzsicsenyi, a bit better. And within Basatanya, we were interested about this pottery style, what is behind of this cultural shift and behind this different pottery styles that appear there. So for that, we analyzed 150 genomes altogether from the Great Hungarian Plain. These are the numbers from the different sites I presented you previously, 22, 30, 30, and 74 from Uzsicsenyi, plus some small sites I won't mention today. And as I said before, sample preparation extraction happened here in Vienna. Capture and shotgun sequencing was performed at the Harvard, and data analysis mostly by me in Budapest with some help from other guys. We did standard population genetic analysis. We used this identical by Basin-based very popular method developed by Harald Ringbauer. And then we also estimated effective population size and inbreeding in our groups. So that would be the first image that I wanted to show you today. It's a principal component analysis. I think you are all familiar with that. This is a standard European layout. We have the hunter-gatherers here, we have a steppe here, we have Caucasus, Anatolia here, and most of the early farmers are concentrated here. And then we divided it into two zoom-in pictures. So first we have the early middle, late Neolithic here and the late Neolithic early Copper Age here over there. We always try to use the chronological abbreviations of the given region, so that might brings you in some confusion, but I hope it's not too much. So basically in the middle Neolithic Polgár area, so the study of Pere Gelabert, we see some variation in the Western hunter-gatherer ancestries. But then in the late copper age, same area, Polgár area, Csőszalom site, it gets concentrated. And in the early copper age, Polgár area remains concentrated also, but 100 kilometers away in Szécsényi sites, we see a great variation. And then we labeled here the outlier individuals. We also calculated, so proved them as outliers on the PCA, and then labeled them. They are mostly females, but not solely females. And checking their grave goods, they are nothing special. So they don't look to be outliers on the site. They do not differ from the others. Okay, so we have two outliers from Basatanya in blue, and the yellows are the Szécsényi outliers. After that, I was interested using this F4 statistics on the diversity further. And since the Szécsényi site was already part of this Lazaridis paper, some samples of it, half of the samples, I wanted to understand whether we really have any Levant-related ancestry there, or whether the diversity of the Eastern European hunter-gatherer ancestry is observable using this F4 statistics. And what we see here that in the Polgár area, no definite trends and no extra sources or influxes are observable with this method. But at Szécsényi, we really have some individuals who are outliers, who have some excess of Eastern ancestry, a little bit shifted toward the Eastern European hunter-gatherers. But I must emphasize that they are all in the negative. They all have negative F4 values, so they all have more shared ancestry with Western hunter-gatherers and the Turkic Epipaleolithic than with the others. Still, when looking at so Pere used the Western hunter-gatherers as a source in his analysis, but when we look at the Hungarian hunter-gatherers and the Iron Gates hunter-gatherers, they are mostly between the Western Europeans and the Eastern European hunter-gatherers. So they are somewhere in between, and we can also try to model with using this QPADM method these individuals as a three-way mixture of Anatolian Neolithic ancestry, Western European hunter-gatherers, and Eastern Europeans. That's what I did. And these were individual QPADM models that I grouped using this box plot. And you see here that it was in their ancestry composition, the Anatolian farmer ancestry was dominant. So it was 80%, over 80% in almost everyone. And we have a little bit of variation between the Western hunter-gatherer and Eastern hunter-gatherer ancestry. And you see in red the Eastern hunter-gatherers, and this is the most in some of the Szécsényi early Copper Age individual. This is also seen here, where we analyze the Western hunter-gatherers and the Eastern separately in this histogram. You see that the Szécsényi has a bit less of Western and more of Eastern hunter-gatherer ancestry compared to the other three important sites that I introduced you before. We had a paper in Nature about Indo-Europeans this year in 2025 together with the Harvard team, and this included a site from Hungary, from Csongrád, Kettős-Halom, which is from the early Copper Age. So basically this is contemporaneous to Szécsényi and contemporaneous to Basatanya sites. And this guy here really looks like, so his genome looks like someone parachuting here from the Volga area. So it really belongs to the Bereznyovka type of ancestry, what he has. And then still, knowing that fact, we must state that we see no robust Eastern influx in the early Copper Age of the area. So we don't see it in the Polgár area, this type of Volga ancestry. But 100 kilometers away at Szécsényi, we have the outliers who can be modeled using samples from this paper of Sandra Penske and Leipzig team from 2023 from the Pietrele or the Varna Copper Age site. So they can be used as sources for some of the samples. If we look at the uniparental genetic composition, we see a great variety among the mitochondrial lineages, and we can state that there is a maternal lineage continuity between the late Neolithic and the early Copper Age of the Polgar area. On the other side, we see significant differences in the paternal lineage composition of the late Neolithic and the early Copper Age. Barsatanya, so this early Copper Age site from the Polgar area shows a dominant lineage, this I2-1A, among many individuals, many males, which also differentiated from the contemporaneous Szécsényi site, which is much more varied and shows a larger variation. Using this identity by descent method of Harald Ringbauer, we built some networks of connected individuals. So these individuals are sharing haplotype segments, and here we apply the filter of at least 12 centimorgan, at least 1 times 12 centimorgan segment shared. And we built this network using a layout called Force Atlas in Gephi, which is a program. And you see the edges are the connections, and they are weighted. So if the total length of the segment shared is larger, then we have a thicker line here, and each node is one individual, one genome. Which is interesting here, that the late Neolithic individuals from the Polgar area are here in the middle of this cluster. Blue ones from the early Copper Age of the Polgar area, Barsatanya, also tightly interconnected. And even a different site from this micro area, this Polgar Nagykasziba is closely there and part of the cluster. And on the other hand, the 100-kilometer apart Szécsényi samples in yellow are scattered around. They do not form a separate and their own cluster within this system. We also had some comparative data from other areas of Europe here, mostly from Croatia and Bohemia, which were available and were high quality enough for these analyses, which requires really good data. If we apply a stricter filtering, so require at least 2 times this 12 centimorgan sharing, our network falls apart. The Szécsényi individuals, they are all scattered in small families, but Barsatanya is still interconnected with the HEAS halom, which is 300 years earlier than Barsatanya. So despite this time gap, they still remained interconnected. And we checked this interconnection between the two sites and established that the guys who connect them are actually not the latest graves of the cemetery. The latest graves are not within these connections. So we think that this connection was in an earlier stage. So probably some part of this community gets separated earlier than the ending and the end of this cemetery. We can use several methods to analyze this network further, and one is this Leiden community detection within the Gephi program, which help us showing whether some communities are really clustered together given certain parameters. And what we see here that yes, the late Neolithic and early Copper Age samples cluster together, and some of the Szécsényi samples also here, although they show a very small clustering coefficient. That's why they are so small in dots. Okay, so several metrics can be analyzed there. And I think one is most interesting among those are these clicks. So the clicks are basically complex subgraphs. So all the nodes, all the individuals are connected in these subgraphs. And when we define these clicks and differentiate between the clicks, the within module, so the clicks within the site, and clicks that connect sites, so between module clicks, and the sexes, then we found something really interesting. So we see different patterns looking at the late Neolithic HEAS halom, for example. We see that females are more rather part of these cliques, and females are rather part of between site cliques. So they are driving these inter-site connections. At Bashatanya, we see that also females are rather part of this inter-site connections, whereas males are rather contributing the inter-site connections. At Szécsényi, we see that both females and males have more between-site connections and less within. So we see different patterns between the three big sites here. Then we went on and tried to estimate the effective population sizes and check for consanguinity or probable inbreeding. Compared to what was published by Pere from Polgár Ferencihat from the Middle Neolithic. So basically, he detected some signals of inbreeding in the Middle Neolithic, but then it disappears in the Late Neolithic. So these two sites have only small signals of smaller population sizes, but no signals of inbreeding. In the early Copper Age, this Romanian-Hungarian border Szécsényi site, sporadic signals appear. So only 3% of the individuals have this signal of marriages of relatives. But at Bashatanya, the early Copper Age Polgár area, it's very striking and also rather rare in ancient data sets what we have here. So strong signals of first and second cousin relationships or level relationships. Okay. We also checked it from the IBD angle, so not just running rams of homozygosity, what was presented here, but check it from the IBD connections and also try to estimate the effective population size using this analysis with the help of Harald. So basically, we see a decline of effective population size in the Polgár area from the late Neolithic to the early Copper Age. You can observe it here. So you have the HEAS connections in purple. Blue ones are the Bashatanya connections, and the green one are the between site connections. So you see that HEAS has in sometimes even less connections within the site than between the two. And these numbers show you the estimated population sizes, and it shows some decrease into the early Copper Age, which means a kind of isolated or closed community in the early Copper Age of the Polgár area. And if we are comparing the two early Copper Age, so the two simultaneous populations, Szécsényi and Bashatanya, just 100 kilometers apart, we see a great difference here. 6.6 times smaller is the population of Bashatanya, effective population, compared to Szécsényi. That's what you can observe here. So at Szécsényi, we have an extensive marriage network, which would explain this, compared to Bashatanya, which is more isolated. Then we went further and analyzed kinship on site level, reconstructing using several relationship estimates. At the late Neolithic Csőszhalom, we only found three small families. The internal connections were relatively weak and no inbreeding, as I told you before. I explained this image already. At Bashatanya, early Copper Age, same area, we see an intensive network of the individuals of the graves with the dominant Y chromosomal lineage of this A2, A1 something, highlighted here in red. So it was found 10 cases out of 16, and 3 kindreds were detected. We call them family A, B, and C. And in family A, we have at least 4 or 5 generations with a dominant Y chromosomal lineage. Family B was interesting because of consanguinity. And what is important here that we have in different colors, we have the different pottery styles, and we see interconnection between the people using different pottery styles. So one family was rather a Tisza Polgár style family. The other family was rather Yes, this apple, the other, the blue, was rather a Bodrokerestur family, but they are interconnected and they have also some distant relatives from the other pottery styles. So it's not that strict as we might imagine it before. And we even have a spruce, a pair of female male here buried close together, so we can state that relatives were buried next or close to each other. And only 2 IBD outliers were found. So only 2 individuals have no connection at all to the others. And they also don't have signals of inbreeding, so they probably come from somewhere else and also didn't have descendants among them. Okay, we checked with the archaeologist Zsuzsa very thoroughly about any kinds of correlation between relatedness, inbreeding, and wealth, so status and burial custom, which is actually very rich in this site, but found really nothing. So that's something that we might gonna understand if we analyze the whole cemetery later on. This site was also studied by Julia Giblin a decade ago for strontium isotopes, and she defined some variability in the Bodrokerestur style graves. So she defined here some non-locals. This is the microregional local range here. She found some non-locals, and then we compared these non-locals with our data and found that some of the non-locals were part of families or got related through their descendants or show some loose IBD connections. But what is more important, that we could state that all the males within the families are also local according to Julia. The other site, 100 kilometers away, Szécsényi, same period, totally different layout. We have small families, 11 small kindreds, buried in large distances. Also, 2 ceramic styles appeared here. They are, in some cases, interconnected. Probably this change from the Salkutsa type, from the Bodrokerestur, happened within 1 generation. So we can even hypothesize which family version would be here the real, because we know that Salkutsa was subsequent to Bodrokerestur, or at least appeared later on. So small families without fathers. So we basically have no father in the cemetery. And we thought a lot with Zsuzsa about possible explanations and models what we can apply here. Various Y chromosome haplogroups appear, but we should tell you, or I should tell you, that the mitochondrial DNA was also very colorful. So the lineages were numerous from the mitochondrial DNA as well. So it's not a clear matrilineal or matrilocal system, but it can happen that the fathers were buried somewhere else. And so basically, we have some radiocarbon data from this site, and based on the Bayesian modeling of Zsuzsa, we think that it was also used for a longer period, so 150, 300 years, which would speak again against of a shorter usage. So we can explain this diversity and these small families with a shorter usage of this site, but then it doesn't fit with the radiocarbon chronology. So basically, the 2 important sites from the early Copper Age to summarize: Vasatanya, we have a patrilocal, patrilineal system. We see abundance of first and second degree marriages, cousin marriages, first and second cousin marriages. It is a highly endogenous community, despite of numerous archaeological connections and trade connections. And this was a cemetery used over a longer period on a lineage basis, we think. But on the other hand, at Szécsényi, it was non-patrilocal, non-patrilineal marriages between kin were uncommon. And it was rather an open community with extensive network connections, or a community used for shorter periods, but radiocarbon speaks against it. Okay, so the key things of this study, we studied we present 125 new individuals spanning from this period. And despite of this profound transformations the archaeologists detect from the turn of the late Neolithic to early Copper Age, we found a significant evidence of continuity in the population, at least in the Polgar area. But despite this continuity, we detect a social transformation. So frequent marriages appear among fourth, fifth degree relatives, and it was probably a socio-cultural isolation as the region was covered with a dense network of small settlements. So it was not geographically isolated at all. And then we see that the social organization and genetic organization varied significantly between contemporaneous sites, so between Basatanya and Szécsényi. And what could be the reasons behind these late Neolithic, early Copper Age changes? It's still a question whether climate, social stratification, epidemics, or some other factors played a role in this. So this is a preprint now that you can read on bioRxiv, and I hope that it appears very soon also in Nature Communications. The main takeaway would be for you that be careful when you try to characterize a culture based on bioarchaeological analysis from one or two sites only. So it always needs to be several sites to come to some general conclusions, and large amount of data, complete cemeteries needs to be analyzed from multiple angles to come to solid conclusions. We continue the ancient DNA research together with Zsuzsa in the frame of this innovation research group, and our aim is to comprehensively analyze this Tisza-Polgár-Basatanya cemetery that I presented you today. So basically sample all the graves available and reconstruct the cemetery generation by generation, and also understand better this chronological sequence of the area from the Middle Neolithic to the Middle Copper Age, and trace, of course, reasons behind these changes that we observed. And that was also a big question of us, whether this contraction in the effective population size, what we observed through the IBD analysis, was really a population decline in the area, or it was rather a social contraction. So that's also a question that we aim to research further. I would like to thank all my collaborators for their great help, and also thank you for your attention.